Hello students, in today's class we are going to learn about mitochondrial structure and function. So mitochondria, they play a critical role in generation of metabolic energy in the eukaryotic cells. They are responsible for synthesis of most of the useful energy derived from breakdown of sugars and lipids, and they convert it into usable form of energy, that is ATP, which is generated by oxidative phosphorylation. Most mitochondrial proteins are synthesized on free cytosolic ribosomes and are imported into the organelle by specific targeting signals. In addition, some of the mitochondrial proteins are also synthesized on the ribosomes present in the mitochondria, encoded by their own DNA, which encodes transfer RNA and rRNA, and some mitochondrial proteins. The assembly of mitochondria thus involves protein encoded by their own genome and translated within the organelle, as well as proteins encoded by the nuclear genome and imported from the cytosol. Coming to the organization and function of mitochondria, mitochondria is surrounded by a double membrane system consisting of the outer and the inner mitochondrial membranes. The inner mitochondrial membrane is highly folded, forming finger-like projections, which are known as cristae, that extend into the interior, which is known as the matrix of the organelle. Each of these components have different role that take place in the mitochondrial respiration process. So the matrix contains the mitochondrial genetic system as well as the enzymes responsible for the central reactions of the oxidative metabolism. The oxidative breakdown of glucose and fatty acids is the principal source of metabolic energy in case of animal cell. The initial stages of glucose metabolism, that is glycolysis, will take place in the cytosol, where glucose is converted to pyruvate. Pyruvate is then imported into the mitochondria, where its complete oxidation takes place to form carbon dioxide, and reducing equivalents are generated, which is used for making ATP. So the pyruvate is initially converted to acetyl-CoA and will then enter into TCA cycle, where it is broken down to carbon dioxide. Fatty acid oxidation also will synthesize acetyl-CoA, which can also enter into the TCA cycle in mitochondria. So the enzymes of the TCA cycle or the citric acid cycle are located within the matrix, and thus they are key players in the oxidative breakdown of both carbohydrates and fatty acids. So the oxidation of acetyl-CoA to carbon dioxide takes place in the mitochondria through citric acid cycle or TCA cycle. The objective of the citric acid cycle is to generate reducing equivalents in the form of FADH2 and NADH by oxidation of acetyl-CoA. So these reducing equivalents such as NADH and FADH2 are then transferred through a series of carriers which are located in the mitochondrial inner membrane during a process which is known as electron transport chain that will ultimately synthesize ATP. The outer mitochondrial membrane is freely permeable to small molecules because of the presence of channels, which is known as porins present within the outer mitochondrial membrane. Now, coming to the aerobic electron transport chain, as you can see, the components of the electron transport chain are actually membrane bound. They are in fact present on the inner mitochondrial membrane, and the objective is to extract protons and electrons from reducing equivalents such as NADH and FADH2 and transport it through an electron transport chain with the objective of making the membrane charged or generating an electrochemical potential, which is ultimately used for synthesizing ATP. So the process will start with complex 1, which is also known as NADH dehydrogenase complex. This binds to NADH and extracts protons and electrons from it. The electrons and protons are transferred to the FMN molecule, and then the electrons are transferred to a iron sulfur protein, through which they are further dumped onto a quinone molecule. During this step, the protons are excluded across the membrane, contributing to generation of a proton motive force. The electrons which are transferred to the quinone pool are completely reduced by acceptance of protons from the cytosol, converting it to QH2 form, which is the completely reduced form. The quinone cycle basically involves shuttling of the quinones from the reduced to the oxidized form, which cycles it continuously in between these forms, which is known as a quinone cycle or the Q cycle. The complex 2 or succinate dehydrogenase complex can similarly extract electrons and will first reduce or generate FADH2, and then from FADH2 the electrons will be accepted and will be directly transferred to the quinone. This complex 2 is not involved in exclusion of protons across the membrane and hence does not contribute to generation of a proton motive force. So from both complex 1 and complex 2, the quinones receive electrons and are completely reduced, which these electrons and protons are then transferred to complex 3, which is also known as cytochrome BC1 complex. And in this process, the quinones will get oxidized again and will be recycled. Cytochrome BC1 complex will transfer the electrons to the series of carriers such as cytochrome BM, BL, iron sulfur cluster, and cytochrome C1 ultimately. Complex 3 is involved in excluding the protons across the membrane again, and the electrons are transferred to a C cytochrome. C cytochrome shuttles between the complex 3 and complex 4 and is involved in dumping the electrons or transporting the electrons from cytochrome BC1 complex to complex 4. Complex 4 is also known as cytochrome C oxidase complex or cytochrome AA3 complex, which is involved in terminal reduction of oxygen to water. For conversion of each half molecule of oxygen to water, 4 H plus ions are used up, out of which 2 H plus ions are used for the actual reduction process and 2 H plus ions are excluded across the membrane by cytochrome AA3 complex in this step. So overall, 8 H plus ions are excluded each cycle of electron transport chain, which leads to generation of a proton motive force. Complex 5 or ATP synthase utilizes this proton motive force for generating ATP. So complex 5 can transport 3 H plus ions back into the cytosol and utilizes this for synthesizing one molecule of ATP or 3 H plus ions that are shuttled across the membrane. The same points are mentioned on this slide for your reference. Next, coming to the genetic system in mitochondria. So the mitochondrial genomes are usually circular DNA molecules like those of the bacteria, and they vary in size from species to species. The human mitochondrial genome encodes about 13 proteins which are involved in the electron transport and oxidative metabolism. For example, cytochrome oxidase subunits, ATP synthetase, NADH dehydrogenase, and so on. In addition, the human mitochondrial DNA also encodes the 12S rRNA, 16S rRNA, and 22 different transfer RNA genes. Mitochondria, coming to the mitochondrial inheritance, they have their own DNA and therefore they can pass the genetic information to the next generation. All the mitochondria are inherited from the egg and therefore the mitochondrial genes show uniparental inheritance, more precisely maternal inheritance, as most of the cytoplasm in the zygote is derived from the egg. Mitochondrial analysis has become a powerful tool for analyzing maternal lineages in humans. Coming to the mitochondrial genetic code, it is slightly different when we compare it with the universal genetic code. These differences are also found in the mitochondrial DNAs of not only vertebrates but also invertebrates, yeast, and protozoa. For example, the codon AGA and AGG encode for amino acid arginine in nuclear genome, while it encodes for stop codon in the vertebrate mitochondria. The human mitochondrial DNA encodes for only 22 transfer RNAs, which are required for the translation process in mitochondria. This is because in humans, uracil present in the anticodon transfer RNA can base pair with all four bases present at the third codon position. This is because of the Wobble's hypothesis that allows base pairing of uracil with all the four bases present on the codon. Mitochondrial DNA are also more prone to mutations because they are not complex with histone proteins, and as a result of which they are continuously exposed to high concentration of reactive oxygen species that are generated within the mitochondria because of the metabolic activities. The mutation in the mitochondrial DNA are responsible for a number of genetic disorders. Now, coming to the similarity between mitochondrial and prokaryotes, the presence of, first of all, they both show presence of double-stranded circular and naked DNA. They both show presence of 70S ribosome. Division of mitochondria as well as prokaryotic cells take place by fission. The inner mitochondrial membrane contains a lipid that is cardiolipin, which is also present in the bacterial plasma membranes. And the folds of the membrane, that is cristae, are considered analogous to the mesosomes that are present in case of prokaryotes. Lastly, coming to the protein import and mitochondrial assembly. Now, in contrast to the RNA components of the mitochondrial translation apparatus, that is rRNA and transfer RNAs, most mitochondrial genomes do not encode the protein that are required for the replication, transcription, and translation process. Instead, these genes are encoded by the nuclear genome, and the protein synthesis will take place in the cytosol, and from cytosol these proteins are imported into the mitochondria for the processes that are taking place in the mitochondria. So we'll look at the protein sorting that takes place in mitochondria. So the mitochondrial protein targeting pathway can be seen diagrammatically, and the proteins that are transported to the mitochondria, they show presence of a pre-sequence which is about 20 to 35 amino acids in length, and it consists of positively charged amino acids. So the first step of protein import into the mitochondria is binding of the pre-sequences to the receptors that are present on the mitochondrial surface and directing them across the transporters. The transporter present in the outer membrane is known as the TOM complex, and the inner membrane is known as the TIM complex, which stands for transporter of outer membrane and transporter of inner membrane respectively. So the proteins are first transferred through the TOM complex, and then they are transported through the TIM complex. The electrochemical potential that is maintained across the inner mitochondrial membrane provides the required energy for translocating the protein. On the cytosolic side of the protein, members of HSP70 family of chaperones maintain the protein in a partially unfolded form that is required while transporting the protein across the outer and the inner membranes. As they cross the inner membrane, the unfolded polypeptide binds to another member of HSP70 family chaperone protein, which is associated with the TIM complex, and it basically acts as a motor protein, importing the protein across the membranes. for this process, it makes use of energy from ATP hydrolysis. Once a protein is imported in the matrix, the three sequences are cleaved off, and then the remaining polypeptide sequence is transported to another chaperone protein known as HSP60 chaperone protein. HSP60 chaperone protein provides an environment wherein the final folding of the protein to the final conformation takes place, which is again an energy requiring process. Coming to the protein sorting to the mitochondrial membranes. So proteins that are targeted for mitochondrial membranes, they contain a sequence which is known as hydrophobic stop transfer sequences that halt the transportation across the outer and the inner mitochondrial membranes. And the protein is, because of the presence of the hydrophobic stop transfer sequences, they are embedded into the membranes, either outer or the inner mitochondrial membrane. Now coming to the protein sorting to the intermembrane space, that is the space between the two membranes. So there are three pathways basically by which the protein can be transported into the inner membrane space. According to the first pathway, the protein will be first transported across the Tom complex, and once transported across the Tom complex, it will stay in the intermembrane space. According to the second pathway, the protein will first be transferred by the Tom complex, and then it will begin to transfer through the TIM complex. But as it is getting transported through the TIM complex, because of the presence of hydrophobic stop transfer sequences, the protein will stay embedded in the membrane itself. Later on, because of the cleavage of the hydrophobic stop transfer sequences, the rest of the protein will undergo folding and will stay in the intermembrane space. The third pathway is by which the protein is first transported through the TOM and then through the TIM complex, by which it will enter into the matrix. Now, once it is in the matrix, the pre-sequences are being cleaved off, and the cleavage of the pre-sequences may sometimes expose the second sequence, which is now the hydrophobic sequence that is again identified by the transporter of the inner membrane or the TIM complex, and it will transport the protein back into the matrix by identifying the hydrophobic signal sequence. So these are the three major pathways by which the proteins are transported to the intermembrane space. So the same points are mentioned on this slide for your reference. And that's it about mitochondrial structure and function. Kindly like, share, and subscribe if you like the contents. Thank you.